Hi, I'm Chandrima Bhattacharya, and today I'll be presenting about genomic drivers for prioritizing candidates during genome mining. I'm from Weill Cornell, and okay, let's start. So in 2014, WHO declared antimicrobial resistance to be a global threat. To quote the then director, he mentioned the world is headed for a post-antibiotic era. But irrespective of that happening, the number of novel antibiotics discovered in the past decade is like significantly less. Since like after early 2000s, we have been in something which is called as the antibiotic discovery void, while there has been a very high rise of like AMR genes and resistomes in the environment as well as those observed in human body. So let us go and talk a bit about the golden age of antibiotic discovery. We see that most of the antibiotics that were discovered during that time came from natural sources like bacteria, like actinobacteria, and other microbes and fungi. So how do we solve this error? One of the best way to do is like using computational technique. One method can be computational genome mining. Computational genome mining usually referred to like predicting the gene that encode a new natural compound of biotechnological interest. That is something that will be showing bioactivity. What do we do during computational genome mining? We usually target regions which are called biosynthetic gene cluster. Biosynthetic gene cluster usually defines a group of building blocks of genes that encode for a specific biosynthetic pathway to produce a specific secondary metabolite. Secondary metabolites usually includes natural products which are produced for like defense, reproduction, and survival of organisms, including bacteria, fungi, etc. Genome mining has been pretty successful to say, like a recent study by Joint Genome Institution found that more than 100,000 BDCs from the Earth microbiome, thus increasing the known BDCs in the IMG-ABC database, which is the class of BDCs, by 31%. And this number happens to be around 54% of the manually curated MyBig dataset. The study characterized BDCs based on location with multiple putative compounds, which can be antibiotics. It has been 10 years since computational genome mining gained popularity, but we still face the same challenges which we did like 50 years back, which led to the decrease in golden era of antibiotic discovery. Namely, validation is extremely tough. It is high cost and requires a lot of time. So how do we validate 104,000 putative BDCs and find for antibiotics? Hence, we devised an experiment to prioritize microbes which can be candidates for drug discovery. What we did is we selected the list provided by the Netherlands Commission of Genetic Modification, or COGEM. COGEM publishes a risk category based on pathogenicity of the organism. Usually, the COGEM list is 0 to 4, 4 being the highest pathogenicity. For bacteria and fungi, the highest pathogenicity is of 3. We downloaded all genomes from NCBI with 3 pathogenicity, and we took equal number of bacteria from risk category 2 and 1 to increase the size of a database. Another reason for using the COGEM species is the fact that they are very well characterized and studied because of the pathogenicity. Irrespective of them being well-studied species, we find that most of the bacteria, that is more than 85% of them, are novel. We identified 566 BDCs from the 57 bacteria. The least number of BDCs per organism was observed was 2, belonging to genuses like Salmonella, Streptococcus, and Francisella. The maximum that we observed was seen in Mycobacterium pseudocystis, who had 29 BDCs. The most characterized BDC was for a strain of Staph aureus, with like 4 out of 6 being characterized. We also repeated the study for fungi, where we identified 617 BDCs, out of which 555 of them are novel. That makes it around 90% of the BDCs identified from fungi to be novel. The least number of BDCs per species was for Rhizopius azureus with 3 BDCs, and the maximum was for Aspergillus Welsvichiae with 68 BDCs. We then tried to see what kind of trends the bacteria and fungi exhibit. This is the relative abundance plot of the type of BDC classes that we observed. For example, we see that bacteria and fungi show unique classes. Fungi has a higher abundance of classes like terpenes and NRPS, while bacteria has an abundance of PKS. We also noticed that there are new classes like aryl pluralin, which is mostly abundant in bacteria. We further studied the genome size, and we saw that genome size had little effect on the number of BDCs observed in fungi, which is very contrast to the more ambiguous finding observed in bacteria. We see a direct correlation of the genome size for bacteria. A previous study suggested that prokaryotes with an average of 2.4 gene cluster per MB usually are microorganisms that are capable of producing a wide range of natural products. With respect to that, we see that the average size observed for bacteria is 1.93 BDCs per MB in our studies, with the highest being for Mycobacterium pseudocystis with 4.7 BDCs per MB. For fungi, it's an average of 0.7985 BDCs per MB. The last two results suggest that bacteria and fungi shows differential way of producing natural products and hence should be studied in different categories. We also studied the effect of GC content and find that GC content strongly correlates with the number of BDCs in both bacteria and fungi. With that, I would like to summarize what we observed in the study. Firstly, genome mining has undoubtedly revolutionized natural product discovery, but there are several means which can help advance the field further, especially with respect to developing new computational tools and experimental techniques to exploit the vast potential of microbial genome. In this work, we highlight the current challenges in genome mining, as well as highlight improved methods to rank and select microbes for prioritization. Interestingly, during the analysis, we also found 2 compounds belonging to RIPS and NRPS, which is conserved in family level, which is usually not observed before. With that, I would like to thank all of you for listening to my presentation, and I would also like to thank the Microbiome COSI, my labs, Mason and Krumsick lab, and my program. Thank you.
Thank you very much for the presentation. We have time for some questions. Okay, so maybe, can you hear me if I speak to this microphone? I'll try to hear. Okay, great. So what do you think are the greatest challenges from a computational nature in finding BCGs?
One of the biggest challenge from computational nature is that there is like what we observe may not be true. So the tools usually can predict like more BDCs or less BDCs based on how they are trained. So the most common tools are things like anti-smash, and their database and the way they are trained has like limited approach based on experimental science. Currently, genome mining is still not as well developed as other fields. So computationally, what you see may not be present, which kind of makes you question if this is correct method or not.